Welcome, everyone. These are my exam 2 explanation slides for the figures I analyzed for my exam 2 research. The first two figures we're going to be discussing are from the paper analyzing E. coli bacterial growth rate in different environments. This figure 3.16, was looking at, or the purpose of this was to determine if the growth in E. coli bacteria was more led by a genetic factor versus an environmental factor. In order to do this, the researchers took different or similar E. coli cell cultures that had been grown in special mediums that allowed for growth and placed them in 65 by 300 millimeter test tubes. These test tubes were immersed in separate baths of 25 degrees Celsius or 30 degrees Celsius. Their rate of growth was calculated and recorded in the first part of this graph you can see. And the growth was measured using spectrophotometers that took recordings of the mass and then dried aliquot masses, which allowed for a determination in cell replication. After the two samples had been placed in these baths for about 2 hours, they were then transferred to the same bath at 37 degrees Celsius. In this bath, they were allowed to grow for another 2 hours with the same methods of measuring that were used. What they found through this experiment was that the change in temperature caused a difference in growth rates when the temperatures were different. That being said, when moved to the same temperature environment, they were able to grow at almost the exact same growth rate. This likely shows that the environment is causing the change in growth rate versus the genetic factors of the E. coli bacteria. They were able to hypothesize that they hypothesized that this change in reaction rate and growth rate was due to chemical reactions and how they occurred slower at a lower temperature than they would at a higher temperature. The second part of this experiment looked at the how different temperature changes affected the growth rate of the E. coli bacteria. This was done through a series of step-up or step-down experiments. This was the same process with putting the E. coli bacteria into different temperature baths and moving them to either a higher or a lower temperature. However, the temperatures that were used were different. As seen in the table, a step-up experiment required the bacteria to be placed in first a lower temperature bath and moved to a bath of 37 degrees Celsius, and a step-down experiment started them in the 37 degrees Celsius bath and moved them to a lower degree Celsius bath. Throughout this experiment, they calculated the specific growth rate at a normal initial temperature, the transient or middle growth rate, and then the growth rate that was normal at the final temperature. And they found that the growth rate changed based on the change in temperature. So what that means is the higher the change in temperature, the greater the change in temperature, the greater the transient growth rate was able to be calculated. And this transient growth rate describes a change in bacteria that can be calculated. And so as you can see, when there's an N, that means that the growth rate didn't really have to make a big adjustment. It was almost able to change right away. And the bacteria didn't have a hard time adjusting versus in a temperature change that is far larger or far more significant, the bacteria took longer to change to that normal final temperature and therefore had a transient growth rate.